Today we have an interesting topic. What do you mean by activation of amino acid and what is a charged tRNA, the first step in protein synthesis or translation? Both these reactions are catalyzed by an amazing enzyme which is called as Aminoacyl tRNA synthetases. This enzyme can literally read the genetic code and can join tRNA with its specific amino acid. Let's move into the detail of this enzyme. This enzyme has three-part active site: an amino acid binding site, an ATP binding site, and a tRNA binding site. Recent reports suggest that there are at least 23 different aminoacyl tRNA synthetases, each one specific for an amino acid. So first step is amino acid activation. Amino acid activation is simply the formation of aminoacyl AMP. Amino acid combines with ATP, forming aminoacyl AMP with the release of pyrophosphate. Let us see the reaction. So this is aminoacyl tRNA synthetase, the enzyme. There is an amino acid valine, then ATP. So this enzyme joins this valine with ATP, forming amino acid AMP or aminoacyl AMP with the release of 2-phosphate or pyrophosphate. So 2 phosphates are lost during this reaction or during this phosphorylation. Now the amino acid is called activated, or amino acid that is bound to AMP is called as activated amino acid, or aminoacyl AMP formation is called as activation of amino acid. The next step is the formation of charged tRNA. So this is called the uncharged tRNA, where the corresponding amino acid is not present. A tRNA that is bounded with its specific amino acid is called as charged tRNA. Second step is the charged tRNA formation, where aminoacyl AMP enzyme binds to tRNA, forming amino acid plus tRNA. Let us see the reaction. So an uncharged tRNA binds to the active site of the enzyme. It has bound to the active site of the enzyme. Now this enzyme transfers this amino acid from aminoacyl AMP, forming aminoacyl tRNA. So in the next step, AMP is released, charged tRNA or tRNA with its corresponding amino acid is also released. This process of joining tRNA with its specific amino acid is called as tRNA charging. Here the use of phosphorylation or ATP is the transfer of high energy bond to this tRNA molecule, and this stored energy is finally used for the formation of peptide bond during translation. So this is the actual bonding. There is tRNA, at the amino acid acceptor arm there is adenine residue, and this is the amino acid. As you can see, amino acid is joined to tRNA through ester linkage. This is the ester linkage to either second position or third hydroxyl group of the adenine residue. Here it is the third position. Amino acid is bonded to the tRNA by ester linkage at the third position of adenine residue. Hope you are clear. Thank you so much for your support. You are with biologyexamsforyou.com. Please consider subscribing this channel.